But there's 4 other things that need to happen. You need to know how fast that piece of DNA decays, because if you only know how that it's there, you don't know how long it's been there, essentially. Then you need to know how far it's come, and that's an oceanographic problem, which needs to be solved. And then ultimately, you need to take those sequences and turn them into information about the organisms. Now, what's really exciting about that, and I know Eric Reyes at Minderoo has done some of this work, is that from some of the metagenome sequences, you can start to see actual metabolic activity in the animals and and plankton that you're sampling, and that is super exciting. There's some wonderful correlations that you can find, but it's really getting from that strand of DNA to the animal. A lot of people have used correlations, so we see a school of fish, and it correlates with this amount of DNA. So those correlations are really where a lot of people are putting their effort. But I think there needs to be maybe more of a theoretical framework and more quantitative framework. I know that's been being worked on in many organizations, but that's, I think, where the challenges are.
Let's continue talking about where people put their energy and focus in this space. Vincent, your research vessel sits proudly in the Dubai harbour at the moment. You're clearly committed to this, this type of work. Where are you putting your energy and focus right now?
Well, eDNA is actually a big focus of the organization for all the reasons that were just summarized. And I think there's a lot of promise in it. And until we iron out those details about how long has it been there, how long will it remain, what are inhibitory factors when you're trying to see, because there's a bunch of technical problems. But the dream is the ability to assess, to some degree, you know, fisheries or to determine diversity or these kind of things. So we're kind of going at it lighter and sort of looking at diversity. And we've actually used it in Maddie, who's here in the audience in the back, as our onboard scientific director. Did a sort of an interesting thing of putting down a CTD normally with the rosette and kind of, there's a word for it which I forget, instead of going up and down with it, just sort of collecting bottles as you move along, looking for changes in eDNA of organisms that might be associated with bottom, not objects, but structures like thermal vents, for example. If you can't find one with traditional methods, can you find a signature of it in the DNA? And that actually Maddie was able to do in the Atlantic Ridge, in just kind of a test experiment where she thought there was something there, and so while they were driving over, they were collecting water along the way. So for us, the overall mission of OceanX is exploration and discovering new organisms, and discovering new biomes and new geological structures. And part of that is to use eDNA to find those things, and also once you find them, to characterize them. So we found a new brine pool in the northern part of the Gulf of Aqaba while we were in the Red Sea, and of course all these mats along the side, all these bacterial mats. So that's not so much eDNA as it is just taking soil samples. But we also took a lot of soil samples with Carlos Duarte, who was working with us, and kind of sectioning them to determine if you can tell, you know, what has been there previously by what they left behind. So all these are ongoing kind of, it's still in the experimental stage, but the promise is very cool.
I'm sure Pri knows all about the experimental stage. She's been working very hard at Minderoo Foundation in kind of developing our eDNA program. Pri, just maybe tell us what you've been up to. You've conducted plenty of expeditions. What have you learned, and what have you identified as some good kind of international collaborations?
Yeah, for sure we face the same challenges that we have just heard from Anya and Vincent. So OceanOmics was actually born of realizing the power and also the challenges of applying genomics tools in the context of ocean conservation. So our work is actually trying to address these major gaps and limitations that are currently preventing the implementation and adoption of eDNA technologies at scale. And this captures multiple sectors, as we just heard. So it captures improvements on how we sample eDNA from seawater across a range of marine environments, from coastal habitats to the deep sea, how we generate relevant sequencing data that contain enough genetic variability to differentiate species and populations, how we translate these raw sequencing data into community composition and abundance estimates, including improvements to taxonomic classification, and population metrics using, in our case, machine learning and AI models. And finally, we are also committed to generating and openly publishing reference genomes for thousands of species of marine vertebrates, which is our group of interest, to be able to accurately identify all the unknown fragments of DNA that we retrieve from seawater. So this particular effort, the creation of this reference genome library, is a key resource for marine genomics and eDNA applications, and one of the major roadblocks for improving and scaling eDNA by monitoring tools. So as we saw in the video, so between the video and the current data, we have already made some progress internally, and there is this current global effort in terms of producing reference genome material to be able to enable genomic applications. But at the moment, we still have a massive lack of reference genomes for marine vertebrates, which sums up to 19,000 species that we have no very incomplete genetic information. So we are working with a number of partners to be able to address these gaps, these limitations. It also captures the work that we do at sea. So a core component of our activities is the execution of research expeditions, which started for us as a proof of concept, where we demonstrated the implementation of our complete eDNA workflow at sea, including high throughput sequencing and bioinformatics in the field. And over the last 3 years, since the establishment of OceanOmics, we have conducted 13 expeditions dedicated to eDNA sampling from areas in the Indo-Pacific that are historically understudied and from sites of high conservation relevance. In our research expeditions, they actually act as the main platform through which we translate the methods and tools that we develop into field applications. So as we continue to improve and refine the tools, the gear, and the methods for a rapid and low-impact monitoring of marine biodiversity and ecosystem condition, we produce these highly valuable baseline biodiversity assessments that can be used to inform how ecosystems respond to environmental change, how they respond to different management strategies, all of those crucial questions that are foundational in the context of marine conservation.
Pri, thank you. You may have spotted some images cycling through. We saw Vincent in action a little earlier. We saw some of Pri's colleagues at the Minderoo lab on board the Pangaea, which you see on the screen. Not quite as impressive in size as the OceanOmics Explorer Valletta. But nevertheless, both ships there doing very, very important work. Ariana, we finally get to you. You'll be pleased to hear I don't think we found any images of you online to share with the audience. But you represent the Australian government here in Dubai, and specifically the partnership between Minderoo Foundation and the Australian government. Can you tell us about that collaboration, and in particular, what potential does Parks Australia see for using this type of technology?
Thanks. And I feel a little like the odd one out on the panel here, surrounded by these experts, but I think it's also really important to share with you how we can see this type of technology and this type of information being applied in a practical context and actually informing government decision making. So the partnership with Minderoo is actually one of several partnerships that were announced under the Australian government's $15 million Ocean Discovery and Restoration Program. And the point of the project is to identify and work with partners to deliver projects that increase our understanding of and ultimately the management of our Australian marine park estate. And this specific project with Minderoo is really about informing our understanding of the potential of eDNA technologies to monitor the health of our marine parks. So the Australian government committed $3.4 million to the project, and Minderoo actually made a co-contribution of $8.4 million, which means that we have an almost $12 million project. This project will run for a number of years, and over the duration of the project, several expeditions will be conducted off the coastline of Western Australia, as well as in our Indian Ocean territories. These voyages will retrieve DNA from water samples and then identify the genetic signature of key species in that water. And we'll be using that data to develop new and efficient methods to monitor the status and trends of key species over time. Minderoo's research vessel, Pangaea Explorer, has been adapted for this purpose and will serve as a primary research base for the expedition. A key project output that we're looking for will be draft guidelines on the application of eDNA to monitor biodiversity in our marine parks. The project will also inform how eDNA could provide a robust and cost-effective ongoing marine ecosystem monitoring tool. So why are we doing this project in the first place? Well, Australia has 60 marine parks covering over 3.5 million square kilometres. You know, this is a huge estate, which means that monitoring its condition, protecting and conserving its natural values, and ensuring that we're supporting ecologically sustainable use of those areas means that we really need to understand what is happening on the ground, or in this case, in the water. We need to be able to understand the state of those ecosystems and also to track that over time, over a very large scale, and some of these areas are incredibly remote. So efficiently monitoring the state of those marine parks is extremely challenging. So that's why we're investing in this project, because we want to understand the potential for genomic testing methods to complement our existing toolkit, to enable the rapid collection and processing of information on the marine biodiversity in our parks, and also understand how eDNA can provide a different perspective, to our more traditional methods around visual and acoustic methodologies.
Well, we'll go into more detail about, the necessity to move at pace, because there's so much we don't know at the moment, and this technology can answer so many questions that currently remain unanswered. Anya, I wanted to talk about kind of the global collaboration efforts, what you've been seeing in your region, and talk about the importance of those collaborations.
Yeah, I think bringing this up to an international level is really important. And just to say, I spent almost 17 years as an engineering professor at University of Western Australia, and the beauty and the untouched, pristine nature of the Western Australian marine area is amazing. Also, when I was there were not marine parks. I think the first one was established, I can't remember exactly when, but it was a place where it was urgently in need of marine parks, and now they're have been enacted, so that's a fabulous thing to see. Internationally, I also co-chair the Global Ocean Observing System, and that's where we have put in place a number. So this is the conversation that we need to have at what we call GOOSE, which is we have a series of essential ocean variables, which we are kind of putting forward for nations to prioritize, and those include biodiversity, essential biodiversity variables. So I think one of the critical pieces of conversation we need to have in all nations, and whether Australia, Canada, where I am, but all nations, is how do we crystallize the application of those essential ocean variables so that all nations can push them forward. And eDNA is a critical tool to get there. It's also an accessible tool, and it can be matched up with things like underwater video and underwater vision profiling to really quantify, or to start to quantify in a correlative sense what it's doing. So it's a huge, huge opportunity here, and its cost is being driven down. So Goose is really focused on trying to bring clarity to what nations need to do to observe their biodiversity. And we have a bioeco panel which would love to talk to all nations about how to build that international framework.
Just picking up on the point, you say it's getting cheaper, more accessible. Could people in this room be conducting their own bit of science? Is there room for citizen science here to kind of contribute to the findings, or is it not quite that easy?
Well, certainly in imaging, it's very much done. So what we do is we put up a whole bunch of images, and we say match your skills against the experts. And then for those that get all of them right, we actually use the data that they're using. So we use that as a criterion by which we choose which of the citizen science identifications we actually put into our database. So there's really interesting and fun ways to get citizen science moving along. I think eDNA per se is highly technical and will always need an expert to do that work well. But I think there's huge opportunities, certainly.
I mean, we did, though, in the Green Pavilion yesterday, we did a demonstration of the MinION, and we ran DNA sequencing in front of everybody. So the process of collecting the data is actually not that difficult, but the big difficult side is on the other side of it, analyzing it and ensuring that it was correctly collected and all those kind of things. So there's a little citizen science in there, but I don't know.
The last time I looked at home, I didn't have a submersible in the garage.
Yeah.
Which is what you'll need if you need to...
That's our goal.
to get some samples deep in the ocean. Tell us a bit about that.
So when we were building the vessel, we thought, and I also should correct that I'm not an oceanographer, so I don't want to sit with I am a neuroscientist, that's my background, but I collect proteins from the ocean for use in neuroscience, but have been doing this work with OceanX for 10 years now, running, sort of managing the program. But when we were building the ship, the thought was that the deep ocean remains the most difficult to get to, and also our founder, Ray, really wanted to have human exploration. And the business has been going in the direction of robotic exploration since Ballard, was pushing ROVs and things like that, and even Woods Hole. That's sort of, they've still got Alvin, but manned submersibles aren't. But we feel strongly that having people see things, and I can tell you on our 1,000th dive of one of our vessels, and every scientist who comes on who doesn't think they're going to want to do it always wants to do it. So human observation for us is important in a vehicle. We have ROVs as well, and for deeper water, but the deep is very inaccessible. And I always say that, to my mind, a lot of the information that we've gathered for climate modeling and for weather forecasting is because we have the huge access to the atmosphere through satellites, through sensor arrays all over. But except for a few notable sensor arrays in the ocean, there just isn't a lot. So if you want to see something in this three-dimensional space, still, you got to go get it. And ships are very expensive and difficult to do, so autonomy needs to come about, smaller, but to date, it's still challenging. So we decided to wrap all that into the ship.
Could we, at some point in the future, have tens of thousands of autonomous vehicles, collecting this data? Because, as I alluded to earlier, the race is on, really, to get the job done. We tend to forget how much ocean there is out there to explore and to take samples from.
One of the, as I see it, technical problems about the ocean is the inability to know where you are underwater. And without, there's no GPS. And so as soon as you go below GPS depth, and you start losing tracking. So to track an AUV underwater and know where it is exactly, it either has to come to the surface or it has to get a reference ping from somebody who knows where they are. So it's not easy to send something down and collect samples and know where it is without it returning to the surface on a regular basis. So we have AUVs and things like this, and they're still not to the point. Now, on the surface, sail drones or things like that, or things that you could run from a drone up and down knowing where you were, I think that's probably going to be but really getting down, what we really do well is collect samples manually by seeing it on an ROV, like ROV drivers do, and from submersibles, so that there's not dragging, which is what has been done historically. So we do much more discrete collection of individual organisms when we go down there, and sequencing of water from around organisms and soil samples and things like that.
You wanted to pick up on that?
Yeah, Vincent, that's a fabulous point about locating yourself in the deep sea. And what's really moving now quickly is sound technologies that are able to echolocate, and some of the other technologies around creating information from data in situ, so you don't actually have to send all the data to shore. You can basically, through some super and semiconductor technologies, if you can get them in situ, you can start to whiz up the data in situ and send that back to shore, which is way, way cheaper than sending long sequence data. So I think some of the communications progress can really help eDNA become a more relevant and easier to technology to use in the deep sea.
Yeah, and we're really hopeful for Starlink satellites because they're low orbit, low power. So, but still the problem of bandwidth in the satellite realm, and sending things out to sea is still not an easy task. It's still a very harsh environment, and underwater is also harsh. And losing such vehicles is expensive, and people steal them and crash them and run over them, and so it's a wild, wild west in the open ocean. So things that, for example, we're excited about tagging animals that will collect information. But then they transmit the position. You got to find the tag. But maybe with low orbit satellites, we'll be able to transmit more data with less energy into the orbital sphere. As long as we get our friend Elon to allow us to use that, that seems to be a real powerful addition to the field.
Ariana, I just wanted to say, first of all, proud to be partnering with Parks Australia on a great project. I just wanted to kind of ask you, is that quite rare to have, government in a relationship with a philanthropic foundation? Is it kind of quite distinct to those relationships with, say, universities or other academic institutions?
So I should say, as the Australian government, we absolutely understand that we need to partner with other organisations if we want to achieve, the outcomes that we're striving towards. That is a given, in any public policy space, really. And whether that be on sea or on land. For us, working with others means that we can leverage their deep expertise. I'm sitting here on this panel just going, wow, these people really know their stuff. That is not government's, area of specialty, right? So for us, it's very important to partner with others to leverage that knowledge, but also the other perspectives that they can bring, the innovative approaches, especially if we want to be at the cutting edge of conservation. And in some cases, let's face it, working with philanthropies means that we can leverage financial co-contributions, which are also incredibly important. So for example, particularly in Australia, we know that we need to work with First Nations people, the traditional owners of our country, our Indigenous people and local communities who have demonstrated effective stewardship of land and sea for millennia. We have a lot of information to learn from them. I think in the case of the partnership with Minderoo, obviously partnering with this philanthropic organisation, we have been able to, tap into the innovation that you guys are developing. Obviously there is that financial capital as well. The access to resources like the Pangaea Explorer. We really see that philanthropies can be quite flexible as well. And in the case of Minderoo, being able to leverage the existing OceanOmics program was obviously a benefit as well. But ultimately we need to partner with a range of organisations, whether that be philanthropies, whether that be academic institutions, First Nations people. So for example, under the Ocean Discovery and Restoration Project, we are also partnering with the Australian Institute of Marine Science and also our Commonwealth Science Agency, CSIRO. Different partners help us address different gaps and different needs, which ultimately are really important for achieving the outcomes that we want. And I think, that's also what we and our partners, that's what we're here about. That's what we're here for, right? Those outcomes. I'll leave it at that.
Perfect. Pri, just so you don't feel left out, we're coming back to you in a minute. And I want to do a little bit of a tease to keep our online audience and people in the room. We'll be telling you shortly about how you can access some of the data that Minderoo has been collecting over expeditions in the last few years. So that's still to come. Bit of a teaser for you. Just want to pick up on the point you make there about having access to organizations with innovative technology that can kind of really help determine the narrative, answer the questions. We've probably all got gadgets and things in our houses that felt great at the time, like the electric carving knife that I think was used once. I also had something that you wrapped a shirt around and steamed it and got rid of the creases, ended up with more creases. Coming to you, Anya, is it possible that there's still some kind of old ways of doing things that we should still harness? Because that is the best way of getting data.
Yeah, absolutely. Still the gold standard for sampling ocean water is to use a CTD cast, which is a conductivity temperature depth rosette, where you send the apparatus down to whatever depth you're interested in, and you close a clean bottle in that space, and you pull the water up to the surface. And so there's ways, and then you do the sequencing carefully in the lab. So there are absolutely simple traditional methods that we're always going to have to use to ground truth. And that's where also our training needs to happen, where we need to make sure that we're not sort of getting ahead of ourselves and saying, sometimes you do see eDNA maybe a little bit used a little bit in vain, if you will, where people are saying, oh yeah, we found all. I'll just give you one example. I was in Western Australia actually sampling for rock lobster larvae. They're very hard to find. They're glass larvae, which you can almost not see when you have them in a tiny bucket. And we were trying to figure out what these guys were eating. And so we were sampling the gut of the larvae and saying, what DNA can we find in the gut of the rock lobster larvae to figure out what they're actually eating? It was a bit of a mystery. anyway, we found out that almost all of them were feasting on these colonial radiolaria, these undersea microbes that are kind of not the food you'd think, and they were all eating them, and they were eating them voraciously. And we thought we had kind of a nature paper in the making. And then I said, hold on, before we go back into the gut, let's just take a little swipe on the outside of the larvae and on the net and make sure that we know what we're doing. Well, turns out these radiolarian were just producing so much external DNA, and when you caught them in a plankton net or in a bottle, that just went everywhere, coated everything, and turned everything into a positive result for radiolaria. So that basically meant that we had to throw out all our big, exciting thinking around and go back to really, really careful, state-of-the-art, how do you do it right techniques. So I think there's times when we just need to get back to basics, to your point.
So, Pri, thank you. Back to you. Let's talk about scaling this eDNA technology, and particularly the application to marine protected areas or fisheries management. What role can it play there?
So I think one of the main motivations, if not the main motivation to deploy eDNA technologies to MPA and fisheries management is its ability to be scalable, to produce robust biodiversity data in a low impact and cost-effective way across both spatial and temporal scales. So the potential is there, and that's what drives Minderoo and OceanOmics to work actively in this space. But to make eDNA technology fully scalable, we need to make them accessible too. So we need to increase our capacity to sample and process eDNA and make those procedures affordable. But we also need to amplify the uptake of eDNA approaches by local, national, and global conservation and fisheries organizations. And that's why we're so proud of this collaboration with Waite Australia and to be able to work with in this direct connection with them. And as we discussed, the expansion of eDNA sampling and sequencing capacity relies heavily on technical improvements and technological developments. At Minderoo, we are currently investing in a number of automated solutions to enable the large-scale deployment of eDNA tools. And that includes development of automated eDNA sampling devices that can be deployed from multiple platforms, including from park ranger and fisheries monitoring vessels. We are also automating sample processing and preparation of samples for sequencing and all analytical pipelines to make those procedures not only scalable and compatible with the large volume of samples and data that we are aiming to produce, but also to make them accurate, consistent, and reproducible. So we hope that by facilitating the deployment of eDNA tools and the production of eDNA-based biodiversity data in a robust way and including the direct involvement of end users, we will also support confidence and this broader uptake of these technologies. And this also taps into the space, another space that we heavily advocate for in this effort to elevate technological innovation, data generation, and community engagement, which is open science. So we are committed to make all methods, tools, pipelines, and data that we produce available as open resources. And through open science, we want to minimize the gap between data producers and data users. And this is because there is no direct benefit of producing loads of biodiversity data if the data are not aligned with management priorities, or if the data don't reach the relevant management bodies and decision policymakers. And having eDNA research and development conducted following the open science principles, this also helps us in this process of building trust, consistency, transparency, and cooperation among researchers, practitioners, and end users.
It sounds like you want to make the data accessible. We want to make the data accessible at Minderoo. Sounds like you've got something in the pipeline you want to tell us about, Pri.
We do. So this is one of the projects we have been working on for a while. So it's called our eDNA dashboard. So through this dashboard, we are making our eDNA data openly available. So besides allowing access to raw and processed data, this tool also facilitates interpretation, interrogation, and is hopefully another step towards improving the delivery of eDNA data to end users for a potential integration of such data into management decisions. So this dashboard will be made available soon through our webpage. I think we have a QR code for people to access as soon that's released. And this will include all supporting information and procedures associated with these data sets. So all the protocols, metadata for field and lab workflows, as well as the bioinformatics and statistical pipelines. So with efforts like these, we want to help make eDNA technologies and the resulting data accessible, trustworthy, reproducible, and ultimately scalable.
Thank you very much, Pri. And for those of you that haven't taken advantage of scanning the QR code, we'll leave it up on the screen for a little bit longer. But thanks to all the panelists. I'd just like to open the floor to questions now, because I'm sure there's some people in the audience itching to kind of ask some difficult, teasing questions about environmental DNA. So who wants to kick us off with a question? Don't all rush at once. We've got about 10 minutes for questions. Who's going to have the opening question to the panel? Because I'm sure you've had enough of me asking questions. Question just at the front here. If you're able just to identify yourself, that would be helpful. Thanks.
Hello, Alain Desbois from Onica Technologies in Canada. We do have a wave-powered desalination solution, and it's kind of buoys standing in the oceans. Our model is to sail water for 15, 20 years. So these platforms will be standing there for that, longevity. How valuable it is for tracking information on biodiversity, having these devices in the water for that period of time, standing about 2 kilometers away from the coasts in the context of biodiversity monitoring and MPAs and stuff.
Who would like to take that?
I think that could be an amazing opportunity. There are a few caveats. I mean, you have to be careful because a lot of those things get biofouled, and as soon as they get biofouled, they start producing their own DNA. So you need to control for that. But having a platform offshore where you could do regular samples, I'm sure with some of the technologies, for example, that Vincent and Priscilla and others have talked about, would be a fabulous way. The cost, though, is not in setting up the sampling, but then in doing the sequencing and turning it into information. So you need to link up with colleagues, and I'm sure you have colleagues in Quebec, Université de Québec à Rimouski, Université Laval. They all have some really great people working in that area, and you could set it up to have that go, the feed go directly to their labs. So absolutely. But you probably want to do some ground truthing, maybe with some underwater imaging. Underwater vision profilers gives you plankton images. Baited cameras give you fish. So there's different ways to ground truth that. You do that all together and you wrap up a pretty nice monitoring tool.
Anything to add there, Vincent?
I think the vision and the AI monitoring, in addition to is also very excellent. So larger animals. So for example, if a bunch of fish swim by and you see them and then you happen to collect water and you get their DNA, it's kind of a nice ground truth. And then if you keep collecting water and the DNA stays and then goes away, then at least you're getting some temporal relationship between seeing something and seeing DNA. So it's easy to do that. Not easy, but it's better to do that if you have a stable station. So there's a ton of things. And if you have plankton samplers and plankton recorders to ground truth that. And I think the fouling thing is an issue. That's an issue. So maybe there's a kind of a differential recording that you could do to kind of eliminate the fouling or something to that effect. Or, there might be ways to do that. No, if you have stable platforms, they're great.
Thank you. Question from the gentleman. We got one here. Sorry, sir. We'll be with you in a second.
Hello. I'm from Maldives, and I was just curious to see whether you guys were active in the area at all. And two, we recently collaborated with Maldives Underwater Initiative, where they do have a lot of cameras on the reef as well. And we came in to kind of help scale up the activities that they were doing already with our core competence, which is Earth observation and remote sensing, continuous monitoring. But if that data layer can be enhanced with eDNA as well. That I think just adds on layer upon layer, and I'm curious to see the explorations. Yeah.
So first of all, anything happening in the Maldives?
I'm no longer in the. Oh, was that is it Priscilla? Did you have something to say?
I think it was a rogue phone in the front row.
No, because to me this is really a Southern Hemisphere issue in a way. I don't know is the answer. I think that it's actually bringing together different types of data is super powerful. So you were mentioning, is it satellite data that you're talking about? So the big danger in satellites in the beautiful clear waters where you are is that the satellite just sees the seafloor. So seeing what's in the water is hard. With the satellite, you need very, very clever algorithms, and very often they don't work very well. Like in Australia, for example, most of the coastal data on chlorophyll is wrong, and it's very, very hard to get those teed up to the clear waters where you see the seafloor. That being said, it means you can see the seafloor. So if that's where your biodiversity that you're interested in, then what do they say in Australia? Bob's your uncle. It's a really good way to go. So you have to be very careful about what you think you're seeing by the satellite, but then yes, it's powerful.
Just want to pick up on that term AI. Should we be trusting of that in this particular field? I don't know, Pri, whether you wanted to kind of pick up on that, or Vincent, but it makes people nervous sometimes when you mention AI. Yeah, how accurate is the picture that you're actually getting as a result?
Just to mention about the application of AI into this genomics space. So we are very excited with this prospect, mostly because with the amount of data we are producing, so amount of genomic data from individual specimens from a variety of species, like learning the variability that can be detected, can be able to can be used to differentiate the species, populations, and hopefully also individuals within species. That's going to be a powerful tool to explore, to be employed, to be able to give us those signals to be able to differentiate and to resolve that level of detailing in the biodiversity space. So we've been looking into AI mostly in that area to be able to find those markers, those signals within a species genome, to be able to differentiate populations and individuals, to help us with this quantitative approach of eDNA that is still so much in its infancy.
Thanks, Priscilla. Question from the gentleman here.
Thank you. Masanori from Ocean Policy Research Institute in Tokyo, Japan. I'm curious about your views on the effectiveness in assessing the biomass volumes by species by this eDNA, how you see this evolutions of eDNA technologies in assessing this biomass volume by species in the future.
So the question is...
I was going to say, don't look at me. I'm not going to take a crack at answering that.
So the question is, how good is eDNA at telling you the biovolume of species? And the answer is pretty bad. You would need to figure out what the relationship is between one particular type of DNA and that animal. And even then, it'll be different when the animal's small and the animal's large. It's going to be shedding potentially different amounts, and there is not really a correlation between a strand of DNA and the biomass of an organism. In fact, from the example I gave before, it's the tiny organisms that sometimes overproduce DNA to the point where they swamp the signal for other things, and it's sometimes hard to pick out. The biggest animals sometimes have the least smallest trace of DNA. But Priscilla might know that from practice. I don't know whether she has a better thought about that.
No, I agree, Anya. So we have there are some models, some demonstrations of correlation between biomass and eDNA detection for particular species, but that varies a lot and cannot be extrapolated to a whole ecosystem analysis. So it's something that, again, we're trying to explore with small scale and validation studies to be able to understand how the system would work, also understanding the impact of the different environmental conditions on on the section of eDNA, as we heard from Anya as well, all the biological factors that affect eDNA to persist in the environment, and how much of that signal it's being lost by eDNA degradation, by transportation to a different place from where it was shared. So that particular correlation between biomass and eDNA presence and detection is still part of our major pieces of work, given the need for that type of resolution.
Thank you. Vincent?
I think at the beginning of the talk, we talked about reference genomes and the importance of collecting those. I think it's kind of and some X Prize-ish. I know that some of X Prize was here earlier. Concept would be the more people who could do. Here he is, our X Prize guy. I think I was just saying. I thought an X Prize idea is the idea of just collecting lots of reference genomes of teleosts and larger animals, and sort of make that our goal. You know, something very bold of doing every large organism in the ocean full genome assembly. It's very difficult to do. But the little guys are one thing, but the bigger guys might be tractable. It's, you go to Indonesia, there's 14,000 fish, but they have COI on a couple of the fish or many of the fish, but they don't have full genome. When you're looking at eDNA, you don't know which piece of the genome you're going to find. So most of it you don't know where it's from because you don't have reference genomes for it. So one of the things we've been doing here, actually in the UAE, in combination with this company G42, which is a big AI company, is first of all getting the reference genomes of the animals fish that are important to them in the area, and then trying to correlate fish eDNA with fish catch, fish from trawling, fish from and try to make that correlation. But then also use it with unguided, with unstructured machine learning to see if it can derive information. My own use of AI in the neuroscience space has sort of overly proven to me that it's incredibly powerful at viewing data sets that you already have a preconceived notion about what's connected to what. And I think AI in that sense is very powerful. And it can always be ground truth and tested. But the fact is we find all the time with data sets that it's very good at predictive work on data sets that one would think are not predictive. So I'm not ready to throw in the towel on a lot of these answers because we think we just don't see correlation. And the last thing I would say is one of the things that was brought up earlier was we're trying to produce 96 well sort of eDNA, rapid collection and processing of massive numbers of samples through a particular area so that we have a really snapshot to try to use that information. Because if you just go down and take one sample, it's not really very good in a three-dimensional space that's moving and stratified and everything. So can we really saturate collection in an area, shotgun sequence the whole thing, and then do you get some meaning out of all that? Then you might find that the DNA is here, here, and here, but not here, here, and here. And that's only if you go everywhere. If you just drop in and grab from here or here. So we're working on sampling methods that don't require collection of water, that are filtration. A lot of people are doing that. But we have subs, and we have RVs, and we have AUVs. We want them all to be collecting all the time in CTDs and see if we can put a high-throughput pathway in for production of libraries. We're not there yet. This is sort of a dream. High-throughput pathway, so it's very, at a technical level, very easy to go from many, many water samples to libraries to genomics.
It's good to have a dream.
It's good to have a dream.
A question from the second row.
Yeah, so you're taking samples in other countries' waters. Do you ever have to worry about IP and the implications?
Always. And I'm glad you bring it up because that is actually the major problem that we suffer from is Nagoya. I don't want to say Nagoya is something you suffer from, but the idea is that the thought is that you're stealing, valuable IP. And we're doing water sampling in efforts to do this kind of habitat analysis, and all the time we're getting concerns. So part of the reason we're sequencing on our vessel at very high throughput is to try to not have to take stuff out. Right. And of course there's CITES, so if we have collection permits we can use it, then we don't have to take it out of the country, we don't have to all this. But main reason is to collect the data in front of everybody, sequence in front of everybody, and then give the country the data while we're there. So part of our agreements, which is important you bring that up, when we sign agreements, we just signed an implementation agreement with Indonesia, was to ensure to them that our goal is not to take it out of the country. So there's open data, But in my business, if something isn't patented in the medical science business, if you don't patent it, it's never going to be any good to humans, right? Because we don't get to make a drug out of it. So putting everything publicly has advantages and disadvantages. If you can use that data to train an AI that becomes valuable, by making it public, you're actually depleting the value of that to a country. Like Indonesia has a massive biodiversity. So if I take all their DNA, I sequence it, and I, in good nature, put it on a public database, then companies in the West take it, ingest it, train models, develop new proteins. That value is never returned to the country. So that's a very sticky situation. They need to do it in their own country. They want to develop their own AI models, develop their own biotech pipeline from that data. So that sort of inhibits the public presentation of data. So it's a sticky wicket. It needs work.
Thanks.
Anya.
Yeah. To that point, in the Global Ocean Observing System, one of the big issues we have, we deal with UNCLOS, the UN Committee for Law of the Sea, and we try to get permission for vessels to go into EEZs of various nations and to keep that free flow of information. Very often that shuts down immediately when you actually take a water sample. It's bad enough when you're just doing temperature and salinity. But when you actually take a water sample, then suddenly you're taking information from somebody's coastal zone, and that can sometimes include human information on pollutants, on bio compounds, and so on. It goes up the next order of magnitude when you're in Canada and Australia, of course, too. But we have a lot of indigenous communities, nothing about us without us. Very, very recent shutdown of a lot of sampling in coastal zones. We had a German vessel, for example, that had all its permits done through the Canadian government, but could not get permitting through the Nunatsiavut government and the Nunavut government to go into. We
couldn't go to Canada.
Exactly, and so, right, and so, and then the question is, who's benefiting from that? But I think the message is there are nations that need to slow the flow of traditional Western science, because they need to bring their indigenous knowledge into that space. And it's a very fraught environment. So I think actually slowing down and stopping is making us ask the right questions. So I don't have anything against that, but totally, we've had conversations with UNCLOS because we've got massive international research vessels moving through these waters, and then suddenly literally have to do a U-turn with a white wake to get out of, some kind of sensitive coastal zone. So it's tricky.
I'm determined not to end on such a depressing note as that. But that is sad, because you'll presumably want to go back to the same locations, repeat the testing. At some point, you'll want to see that policy is having some sort of impact, and it's showing through the results of what you're analyzing.
I mean, I don't think it's sad. I think what it is simply waiting for the capacity to be built up in those communities for them to take charge of this work themselves. And so that's what our job is then, is to say, okay, who's going to step up? And obviously, it's not going to be them paying for it, right? If we want to work in those areas, we need to increase the capacity of those communities.
We're nearly out of time. Let's just end. We'll go along the panel. What are you excited most about? Anya, starting with you.
Statistics. So I've seen some of the most amazing work done. Colleagues at University of Chicago that we're working with are just blasting through absolutely new correlations between there's and AI. I think, to me, those are the big frontiers, and they're moving quickly. Super exciting.
Statistics, gosh, I'm not sure it does it for all of us. I took myself back to university during the pandemic, and I had a book, Statistics Without Tears. Right. Ariana.
I was so excited to hear you say statistics. That was my undergraduate major, close to my heart. I think from my perspective, good information is the basis of all good decision making. And that's not only at the level of government as well. A lot of my day job is about how do we work with the private sector to get them to recognise value and invest in nature. And the first thing that they always come back to us with is, we need information, we need data. And then once you start talking about the ocean, they throw their hands up in the air in desperation. But I think, there are industries like fishing and in Australia there's a big debate at the moment around offshore wind and renewable energy there. So I think, it's not just for government, it's also for the private sector, and making these better decisions that can ultimately lead to better outcomes for the state of our ocean.
Vincent.
So we are excited about building a system on our vessel that we think is applicable to other places. We should to take genomic data, combine it with all of this oceanographic data that we collect, weather data, underwater, bathymetry, all the data that a boat like ours collects. And we're building a platform here with this G42 company here in the UAE to build a platform to allow all that data to be analyzed using very sophisticated AIs. And that excitement to me is it's a statistical method in some way in AI, but a method of deriving information from very multidimensional data sets that in medicine, we've seen this a lot, that data sets that we don't understand the correlation, AIs can drive information out of data which seems to be too dense and too multidimensional. So that excites me. Again, it has to be ground truth, but we've found already in medicine clear knowledge, clear learning, not just correlation, but learning that we go back and prove is interactive.
Thank you. And finally, Priscilla.
So I'm very excited to see this increasing commitment and also the intentional application of breakthrough novel technologies into the conservation space. So to see technologies that come from magical science that have been applied to human health, now being applied to ecosystem health, it's something very, very exciting to see. So yeah, I'm looking forward to more and more sequencing data to be able to use that sequencing capacity to expand our reference genomes, to expand our eDNA sample collection and data generation efforts. So yeah, exciting times to come.
Thank you. I think we're almost out of time. A few thank yous. First of all, to the organisers and the supporting partners of the Ocean Pavilion for creating this wonderful space for us to use. There's been some fantastic sessions. I hope you enjoyed this one. There's many more to come. If you wanted to kind of plan the rest of your stay at COP, there's always a willing volunteer at the reception desk that will kind of guide you to the rest of the sessions. But we're kind of proud to be a supporting partner, Minderoo Foundation. And I'd also like to thank our panelists today, Anya, Ariana, Vincent, and Priscilla, joining us all the way from Brazil. And I have to say, this Zoom connection has been better than most of the ones that we have when we're speaking in meetings at Minderoo. But it's great to have you all. I think it's been a really fruitful discussion. And have a good rest of your time and a safe one here in Dubai. Thank you.